All right. We're going to talk about synthetic pathways now, and this will be the last thing that we talk about in this unit of study. Right? So far we've been talking a lot about how molecules are broken down, how glucose is broken down, or fats are broken down, so on and so forth. But now I want to talk about just a couple of different pathways by which things are made. All of these are endergonic, meaning that we're storing energy in the final product rather than releasing the energy. And we've already covered protein synthesis in quite a bit of detail. The first one that I just want to really briefly mention is glycogen synthesis. Glycogen synthesis is essentially glycogenolysis in reverse. We mentioned briefly last time how glycogen is broken down, and essentially glycogen synthesis is a reversal of those reactions. Glycogen is the primary way in which we store carbohydrates. Most of it is in our liver. We do store a little bit in our skeletal muscles as well. And in order to metabolize it, we have to break it down, right? And that's glycogenolysis. But storing it as glycogen is a relatively simple process. Large glycogen molecules are somewhere on the order of about 55,000 glucose molecules. So, we're looking at relatively large molecules here, but they do vary quite a bit. There's no defined size of glycogen. All right. The next pathway that I want to talk about is one called gluconeogenesis. Gluconeogenesis. Gluco is sugar, neo is new, and genesis means the creation of. So gluconeogenesis is literally the creation of new sugar. Right? One of the problems that we face in metabolism is that we do use a lot of glucose. I spent a little bit of time in the last lecture talking about the importance of fats in our lives, but glucose is still a really important substrate. Okay. In fact, for some tissues, it's the only substrate that can be used. Your nervous system, for instance, your brain, is only going to be able to metabolize glucose. It cannot metabolize fat or other sources. Okay, so sometimes it's the only source of energy. We'll remind you that that's happening in our brain. Okay, so let's just say that you are fasted, you're starving, or something like that. You use all of your glucose stores. What are you going to do for fuel now? Well, unfortunately, the only option is, if you don't have energy coming in the form of food, is you've got to start creating sugar from other things. And that's where gluconeogenesis comes into play. Okay? So what we see with gluconeogenesis is that amino acids and lipids can be intermediaries for new glucose. Or precursors for new glucose. Okay, they can be precursors for new glucose. The process is relatively simple. Okay. Let's go and take a look at the process. We'll come back to that note here in just a second. So if we take what we've been learning about, and we take pyruvate, for instance, we know that we can convert a lot of our fuel sources back into pyruvate. So if we start with that, we know that we can make pyruvate from amino acids, right? We talked about how during the deamination process, sometimes that organic acid that we get is pyruvic acid. We also know that we can take lactate and we can make pyruvate from that too. We're going to take a little bit more look at that specifically here in just a couple of minutes. All right. We know that if we reverse what we've been learning about in this unit of study, pyruvate can be converted back into glucose 6-phosphate. Remember, that's the first intermediary in glycolysis. And I'm bringing this up for an important reason here in just a minute, and then that can be converted back to glucose. So we see something that sort of looks like that. All right. Along the way, we can convert other substrates back into this. There are some amino acids that are converted to intermediaries of this process, so they would enter right here. Glycerol, which is from fats, is often going to be converted back into one of these intermediaries as well that will ultimately be converted to glucose 6-phosphate. All right? And then we see that glucose 6-phosphate can be converted back into glucose. But here is something that's really interesting about this process, right? This step right here, the one where we convert glucose 6-phosphate to glucose, this step can only be performed, or the step only happens in the liver and in the kidneys. Okay, can only take place in the liver and kidneys. So gluconeogenesis can only happen in the liver and kidneys. It can't happen in any other tissue in the body. But we can ultimately produce glucose, and that glucose can then diffuse out of these kidney and liver cells into the bloodstream, and it can be sent to the brain and other tissues where it can be used. So this is very generally what this process sort of looks like. A couple of notes about this really quickly before we leave this process behind, okay, is that this is not something that we do on a normal basis, right? So gluconeogenesis is not super common. You're probably not going to do it today because you're eating food, right? And as you do that, you're getting the glucose that you need. So you don't need to rely on this. But we do see this taking place during fasting. We see it taking place during starvation. We can see it taking place in people who have really low-carb diets. If you aren't eating sugar, you're going to make it from something else. Right? So we see it in these sorts of conditions as well. We can see this happening during intense exercise. If you are using your carbohydrate stores and you're not refueling, you're going to start making carbohydrates from other sources. So we can see it happening then as well. We can also see this strangely during high blood glucose episodes in diabetics. High blood glucose seems a little bit counterintuitive, but what's happening in a diabetic who has high blood glucose is there's no insulin present to bring that glucose into the cell. So the cell is cranking out, doing what it does, and it's running out of sugar. So what's it going to do? It's going to start making glucose from other sources so that it can survive. So even though there's a lot of glucose in the blood and the blood glucose levels are really high, there's no insulin to bring it into the cell, and as a result of that, they can go into gluconeogenesis. One of the byproducts of gluconeogenesis very often are things called ketone bodies. And a person who's doing a lot of this can go into a state that we call ketoacidosis. All right? Ketone bodies often can be detected in the urine. So later on this semester, we're going to be doing a urinalysis where you're going to look for the presence of ketone bodies in your own urine. But those ketone bodies can also appear in other strange places as well. One of the things that we do sometimes is we give them off in our breath. So somebody who's doing a lot of this, you can actually start to smell the ketone bodies on their breath. They smell very sweet, and the smell of their breath is going to change as a result of it. Unfortunately, that's often a tip-off for families of people who are dealing with anorexia. They'll start to notice that change in their breath because the person isn't eating. They're essentially on a very low-carb diet, and as a result of that, they're doing a lot of gluconeogenesis to sort of keep up. There's one other thing that is worth mentioning here, sort of from a clinical side, and that's that this glucose that's being made can diffuse out of the cells that it's made in, into the bloodstream, and it can be used elsewhere in the body. However, glucose 6-phosphate cannot diffuse out of cells. There are no glucose 6-phosphate transporters on cell membranes. We just simply don't have them. So even though this can be used to essentially go in the reverse direction, and we saw that a little while ago, to make ATP, it can't be shipped out of the cell and go somewhere else. If you were trying to control blood glucose levels in, say, a diabetic patient, one of the things that you might want to do is you might want to prevent the diffusion of this glucose out into the bloodstream to help keep blood glucose levels in check. So in other words, what you might want to do is you might want to block this step. There's a drug that a lot of diabetics are given to do just that called metformin. And metformin is going to block the conversion of glucose 6-phosphate to glucose. That forces the glucose 6-phosphate to stay in the cell and be metabolized, and it prevents the rise in blood glucose levels, so it helps to control a diabetic's blood glucose level. So kind of some interesting clinical applications of this as well. Okay. Just a couple of more things that I want to talk about. We're getting really close to the end. And one of the things that I want to talk about is relating to gluconeogenesis, but I want to talk specifically about how we get rid of lactate. All right? We've been talking about the production of lactate, how it hinders exercise and performance and things like that, and how we get rid of it. If you do any exercise at all, you've probably felt the buildup of lactate in your muscles. You know that it makes you feel really crummy for a couple of hours after exercise. You may have even heard that you have to, quote, repay the oxygen debt in order to get rid of that lactate. But how exactly do we do that? Well, we do it via a metabolic process called the Cori cycle. So I want to take a really quick look at the Cori cycle. Now, part of it is one that we've looked at. So let's just assume here for a minute that we have a muscle. Okay, this is our muscle cell right here. And in this muscle, we are exercising intensely. And as a result of that, we have glucose getting converted to 2-pyruvate, and those 2-pyruvate are getting converted to lactic acid. Right? So that's happening here. this lactic acid is building up in our muscle, and our cell membranes are really permeable to lactic acid, so that lactate is going to diffuse into the bloodstream fairly quickly. So when you're exercising really intensely, you usually see a rise in blood lactate levels as a result of that. So lactate is diffusing into the bloodstream, and it's going to go to the liver, right? So here's our liver right here. And the lactate is going to travel in the bloodstream to the liver. In the liver, okay, what we're going to see is we're going to see the 2-lactate being converted ultimately back into glucose. Okay, this happens in the presence of oxygen, so oxygen is required for this to occur. Okay, this is gluconeogenesis. This is what we just sort of described in the last little bit of this unit that we're looking at here. And then that glucose, because it is permeable to cell membranes, can diffuse back into the muscles to do work. So we complete our cycle as a result of that. All right. Now, when glucose is converted to lactate, we know that one of the things that we're producing is 2-ATP, right? We're producing 2-ATP there. The Cori cycle is going to, or the gluconeogenesis part is going to require 6-ATP, all right? So doing this, there is a net energy loss. It comes out to about 4 ATP per glucose. So as a result of that, this is typically not a sustainable cycle, not something that we can do for a long term, but it does help us to clear that lactate from our blood and from our muscles, and it does help us to return to a normal pH after exercise has ceased. All right, and this cycle does tend to work the best after exercise has ceased. Okay, the final thing that I want to talk about in this unit is sort of a variation on this theme of things that we've been talking about. I want to talk specifically about a tissue called brown adipose tissue. Brown adipose tissue is something that you and I don't have a lot of. The adipose tissue that we have, the fat that we have, is often called white fat or yellow fat. But brown adipose tissue does have a distinctly brown color to it. It's found primarily in small mammals, so squirrels and mice and things like that have brown adipose tissue. Even babies, infant humans, have brown adipose tissue. And these small animals face a challenge that you and I don't normally face, right? Small animals have a high surface area to volume ratio. Okay? What this high surface area to volume ratio does is it increases the rate at which they lose heat to the environment. To put it another way, small animals have a harder time staying warm than large animals do. And as a result of that, they need to generate heat in ways that you and I don't necessarily have to. How do you generate heat? If you walk into a cold classroom or something like that, what you do is you simply start to shiver. And those shivering muscles, those rapidly contracting muscles, are going to generate the heat that you need in order to survive. And small animals do that, but they also need a little bit of help. And that's why they have brown adipose tissue. Brown adipose is fat tissue that is just jam-packed full of mitochondria. So there's a high mito density in brown adipose tissue. So brown adipose tissue is BAT. Okay. a high mitochondrial density in brown adipose tissue. That high mitochondrial density allows us to metabolize the fat at a very high rate. The other thing that makes these mitochondria interesting is that they have very little ATP synthase. What those mitochondria have instead is they have a protein called thermogenin. The idea behind thermogenin is identical to ATP synthase. It provides a pathway for the proton gradient to be dissipated. However, when protons pass through thermogenin, the result is heat. Okay, so a lot of heat is generated as a result of those protons flowing through this protein called thermogenin. And as a result of that, we end up with what we call non-shivering thermogenesis. Okay, so animals, small animals like mice and rats and gerbils and babies and things like that, have this non-shivering thermogenesis that allows them to generate a lot of heat in their mitochondria. And you and I don't necessarily have that. Brown adipose tissue is brown because it has a really rich blood supply. A lot of capillaries in it supplying a lot of oxygen and things like that for this process to occur. And as a result of that, it has a much different color. So there you have it. We have our unit on metabolism. So if you have any questions, let me know. Don't do drugs, and we'll see you in class.